through the small intestine, for example, if you imagine your toothpaste and female reproductive systems, biological males and biological females, meaning that males generate these natural occurring hormones and sex organs, which are produced by X and Y chromosomes and XX. And I'm not going to test you on this, but I'm just trying to clearly define exactly what male and female will be defined in our class. I know that there's a lot of cultural debate about all this, but we are thinking simply from a biological, male has XY, female has XX, and they lead to different reproductive systems. All right. And so at a glance, one thing that we want to note about the reproductive system in males is that males produce their sperm, the cells that can cause pregnancy or they look for eggs, and they are located and made in the testes. When males undergo climax during sexual reproduction, the actual sperm cells will travel out, and you'd think, right, that, just by the basis of location, that somehow it would go from testes and right out the urethra and exit, but that is not the case. Rather, the testes will then release sperm. It'll go through something we call a duct system, which is just like what you imagine an air duct system is, like a network of tunnels for the air conditioning and the heater. And so you can see that it travels around the urinary bladder. This is our urinary bladder that stores urine. Here's a ureter that's been cut, so urine entered into this urinary bladder, right? And it's stored and expands accordingly. But again, our sperm is traveling around the urinary bladder, wraps around the posterior end of that urinary bladder, and then finally will exit out the urethra. Last week we briefly talked about the urethra, and that there's a urethra that shares between the urinary system and the reproductive system. So sperm will take the pathway coming from that duct system that I just highlighted. And you can see this little segment here. This comes from the urinary bladder. And so urine will come through into the same urethra, and so will sperm. So you can see that the urethra is shared between the reproductive system and the urinary system in males. And along the way, there's going to be something that we're going to pick up, we're going to see in a second, that what actually comes out of the testes is sperm plus semen. And sperm makes about 10% of the content. So in other words, when males undergo orgasm or climax, they release 10% sperm and about 90% semen. So now what that means is that this doesn't fertilize anything, semen itself, but it serves as an assistive material to help sperm survive its journey to the outside world. Because there's a lot of things that it has to encounter that can damage it. For example, we understand now that the urethra carries urine through the same pathway, and that urine is very acidic. And so sperm dies when it's exposed to that acidic environment. And so that's what semen is for, is to help protect it from that acidic environment and also the acidity of the female vagina as well. And so try to keep this context in mind, that there's only really 3 topics to learn: where sperm is made, and what is the pathway to the outside world, and also what does it require to become successful in finding an oocyte. And that's going to be, how does it get semen? And it's really simple when you think about it just from a simplistic perspective, that all of this is just sperm traveling out, and then eventually it reaches a checkpoint. As you can see, there's a little structure here that sort of meets that little pathway. There's another one, the prostate gland. We'll talk about these in detail. I just want you to see the overall view. And then one more thing that helps out, and then it helps sperm. It picked up semen, and now it's ready to come outside. So it's really about how sperm gets to the outside world and all the checkpoints that are required for it to actually be successful. And so starting in the testes, if we look at the testes very closely, you can see that the outside layer, the scrotum, is going to be what we've always colloquially called as the sac, the testicular sac, but it's called the scrotum. And this is skin, as you've learned about thin skin in your when we learned about the epidermis. And then there's going to be some underlying unique muscles, such as the dartos muscle. And these muscles play a role in temperature regulation. I think we all have heard jokes or somewhere along the line that what happens to the testes when someone goes or is very cold and they go in cold water? What happens? They shrink. And then when it gets really hot, what happens? They sort of shrivel and go out, right? And that there's a reason for this, because in this space where the sperm cells are made, they are very temperature dependent. They require a particular temperature, and the inside of the body is too hot. And so the testes, a very precious part of reproduction, a very precious organ, is on the outside. I don't know if you've ever wondered, like, why would something so important be outside? And the answer is temperature regulation. And so this dartos muscle is responsible for letting the testes leave the center of the body, or they're brought into the body's warmth in order to regulate the temperature for successful sperm production. And then we have a little bit of a layer of connective tissue that makes finalizes that bit of that scrotum. Inside of this space, right, this is going to be considered your scrotal cavity, this space inside of the scrotum. And then finally we made it to the inside where we have our seminiferous tubules. Seminiferous tubules. This is the site of sperm production. These seminiferous tubules will release sperm as they're being produced, and then they're going to place it, if you're just drawing your eyes to just the purple lines here, these are tubes, and that have, like, they're hollow on the inside, and sperm will make its way out and finally make it to what we refer to as the rete testes, which is only labeled on this side for some reason. Nope, it's not labeled anywhere. Oh, here it is, yes. And this label, what I want you to do is that this label is improper. It actually should be here. It should be going a little bit higher up. So let's draw those labels in. Let's use red. So the rete testes is going to be this site here, where all the sperm cells, when they're released from the seminiferous tubules, they will arrive at the rete testes. Now, the first pathway for sperm to exit will be the epididymis, and it'll also be the site where they develop their tails. The rete testes is just this centralized hub where all these sperm cells will exit from this segment, and they all just pool together in that segment. And epididymis is going to be where they develop their tails, and again, during male climax, this is the first site from which they are released. Do not worry about efferent ductule. Do not worry about straight tubule. We will need to worry about this, but this is the worst picture possible to ask or any of this, but just keep in mind it's a word we need to know. And the last thing to add is in this segment here, there is a layer of connective tissue, one on the inside surrounding the testes. That's going to be our tunica albuginea. That's going to be surrounding the rete testes and the testes, or seminiferous tubules, tunica albuginea. And tunica vaginalis is going to be the boundary outside of where our epididymis is and the scrotal cavity, tunica vaginalis. And these words will come up again in better pictures, like dartos muscle and cremaster. We'll see it again in a second. And the last thing to add is within our tunica albuginea, in this space that surrounds the seminiferous tubules, you'll notice that the seminiferous tubules are collected within their own mini lobes, hence why we refer to those little lobes as lobules. Lobules. Now, the duct system, and I'm going to go ahead and talk about it now. Let me see what picture I got here. Something as we progress forward is our duct system. In other words, how does sperm, what is the pipes that it uses to exit to the outside world? And it got a little bit cut off. And so what we're going to do is add numbers here. We're going to add epididymis for number 1. We're going to add ductus deferens for number 2. And when you're looking at number 1 and number 2, I want you to add vas in parentheses here. Vas. And then number 3 is going to be our ejaculatory duct. And number 4 is going to be our urethra. So this essentially is a flow chart you can make from number 1 to 2 to 3 and 3 to 4. And so that's how sperm comes to or exits out into the outside world. And so it starts at the testes, goes to the epididymis. Follow along. This is our ductus deferens / vas deferens. Now, some of you may understand now why it's called a vasectomy. And that's because this is the tube that they're cutting. They're clipping it on both ends, and so sperm never can really exit out of the testes and come out to the outside world because this ductus deferens is cut. And so this ductus deferens will wrap around the urinary bladder, and then you'll find the seminal gland. And you'll notice that this is under accessory glands. These here are your ducts, your duct system. Duct system. Meaning it's just the pathway that sperm uses to go to the outside world, but along that way, it has to reach certain checkpoints. You can see the seminal gland exists right here, right by the ejaculatory duct. And so when sperm arrives here, this will add some semen to the sperm cells, and it'll help them survive their path or their journey to the outside world. And then the prostate gland is our next gland, which will also be where the ejaculatory duct is. And so we'll also contribute some alkaline mucus and some hormones that will help stimulate the female uterus to sort of draw sperm into it. And we'll talk about that in a little bit. But otherwise, the prostate gland contributes to the success of sperm a little bit more. So you can see sperm travels. It gets helped by the seminal gland, and then it gets helped by the prostate gland as it's traveling out. And then finally, there's one more gland called the bulbourethral gland, and this gland doesn't really have anything to do with semen production. Actually, it does, but for our class, it doesn't matter. For our class, the role of the bulbourethral gland is to provide lubrication to the head of the penis for males when they're aroused. The bulbourethral gland is what provides the lubrication for sexual reproduction. And so that's with the bulbourethral gland. So these are accessory glands. They're not part of the duct system. They contribute to the success of sperm on their way out, but the accessory glands contribute contents to help them be successful. All right, let's move on. And so these are some of those same terms that we discussed, like the rete testes. It's where all the seminiferous tubules merge and sperm before it goes to the epididymis, which is the first part of that duct system. And within there, we have our site of sperm production within lobules. Efferent ducteals, I told you to cross out, but it means going away. So any ducts leaving those actual lobules are going to be considered efferent, but again, this is something you can go ahead and remove. Okay, you do not need to worry for our exam. The scrotal cavities is what's going to house the testes, all in general. The septum, do not worry about this either. But just keep in mind that your testes can be divided into a left and right. So if you look at this midsection here, this is what we mean by a left and a right. But you will not have to worry about it for exam. I'm trying to limit the material you need for the exam since it's coming up. And then we have our dartos muscle. This one we do have to know. This is the one that elevates the testes. And when it relaxes, it allows it to sort of sag away from the body. And the cremaster is another muscle that we're going to see here on this segment here. The dartos and cremaster, as far as you're concerned, do the same thing. They help regulate the temperature by either bringing it towards the body or relaxing and allowing the testes to go away from the body. The difference is their location. The dartos muscle is located within that scrotum, within the scrotum. And the cremaster is completely wrapped around the testes and is anchored to something we refer to as the inguinal ring and the inguinal ligament, which keeps it anchored to the pelvic cavity. So that's the difference between the two, but their role is fundamentally the same, is to help regulate the temperature of the testes. Now, our spermatic cord. Our sperm cells, they need resources like every other cell in our body. They need lipids, they need proteins, they need carbohydrates, they need nucleic acids. And so whenever the small intestines absorb all of those nutrients, they're going to be delivered to the testes to something we call the spermatic cord. The spermatic cord is the essential pipeline for the nutrients to arrive right through the arteries. And also you'll notice that these veins have extensive branching. These veins themselves don't only just carry nutrients to and from the actual testes, but their role is also temperature regulation, that they can actually absorb some of that heat. And so it's referred to as venous plexus. And so they actually do help with temperature regulation. And so that makes up part of your spermatic cord. This Thursday, we're going to learn about nerves and nervous tissue. And so we would also need nerves to make sure that, if predators or anything is biting something very important, you would know. And it's a way of keeping the reproductive organs safe, right? You need to be able to conscious of feeling there. Then we have our ductus deferens, which was one of those pathways that I also called it the vas deferens. And because, again, this is going to be the pathway, the epididymis is here. If you look at your testes, epididymis is that first place where they develop their tails. And during male climax, this is where they're stored and then finally released through the ductus deferens. And keep in mind that our ductus deferens has to wrap around the urinary bladder. And so it's going to utilize the space of the spermatic cord in order to rise from the testes and go up around that urinary bladder, to wrap around that urinary bladder. So spermatic cord, again, is going to be what contains all of those structures. And the last thing to mention, that's really it. There really is nothing else. This ring, I mentioned a little bit earlier that this cremaster muscle is actually anchored to it, but that's incorrect. It's actually a passageway for the cremaster to actually go and connect somewhere else. So it's just an opening that allows for pretty much all of the spermatic cord to go through. Just an opening. And so re-examining our testes, remember here we have our seminiferous tubules within their lobules. And we're going to take a closer look at the inside of those in a second. But right now we're just keeping everything sorted. We're going from large to small. And so as sperm traverses out through into the rete testes, which again, these are the rete testes. The other picture was labeled incorrectly. This is going to be a site where they all pull together. They all pull into that space. It's reminiscent of the word renal pelvis. Remember that you have your major calyx and they go into the pelvis, all of them. And so all the rete testes is sort of a site where all the sperm will pool. And then now our sperm has finally going to go into that first duct system, which is going to be the epididymis. And so this epididymis wraps around the testes, and it has a head, a body, and a tail. Simply, first part, let me get a marker. First part, head, and then body. And then the last bit is going to be tail. So here's our midsection. Let me use different colors. Head, red is body, and then finally you have tail, a tail at the epididymis tail. So those are the three segments, head, body, and tail. Pretty easy there, right? And so it is considered the start of the reproductive system because this is where sperm is stored and it finalizes its maturation and stored until it finally is supposed to do its duty of leaving the system. And so, anything I want to add here? This coil tube is just trying to describe its description, but it always just sort of looks to me like a I know it sounds silly, but like an earbud. I used to have these earbuds that wrap around the entire ear, and that's why I always remember it that way. But it sort of wraps around the testes. So this is just trying to give a verbal description. And so they tend to be on the posterior segment. So as you go to lab, you'll notice that a lot of these terms are the exact same terms that you need to know for your lab final, the exact same material. And so when you look at the models, you'll notice that this is on a posterior end and tends to have a different color on your models. So be conscious of that moving forward. And inside of that epididymis, we have the mucosa layer, or that first inner lining where the sperm are in contact with. They're going to have pseudostratified columnar epithelia. And try to keep in mind that pseudostratified columnar epithelia, they're probably going to have cilia. And our sperm cells aren't always kicking like tadpoles. Like, when we imagine, I have a picture of a sperm cell. If you've never seen one, this is what a sperm cell looks like when it's fully mature. And so it has to, like a tadpole, sort of stroke this tail in order to actually move around or move forward. And it requires a little bit of maturation. They're not all, they don't start off that way. And so they need the help of cilia, stereocilia, to help them move before they've matured. And I'll even show you, here's like a little slightly immature, the tail's not as long. If it was up to me to draw this, I would kind of get rid of this tail to not show it because they don't develop until the epididymis. But you can see that most cells would resemble like something like this. This is even more closer to what they would resemble instead of this, but they wouldn't really have their tails just yet. And so if they don't have their tails just yet, they require the use of pseudostratified columnar epithelia with stereocilia or cilia to just help move those sperm along the way. And again, this is where they fully mature, and that's why I wrote this is where they fully develop their tails. The ductus deferens, which is the next part, right? Because after we've gone through this, the epididymis, right, we traveled down to the tail, and then we finally made our way into the ductus deferens. So it's going to travel upwards. You can see there's no more squiggly lines on this ductus deferens, and it's going to make a long journey around the urinary bladder. And again, sometimes, or traditionally, it was called the vas deferens, and it transports sperm from epididymis to the next site. And this is where we primarily store fully mature sperm. Fully mature sperm. It's already there. I'm not going to write anything. Now, the way I separated the notes is that we know we go from our ductus deferens, and then I said afterwards that we're going to go to our ejaculatory duct. And so, but what I want to do before we get to these sites is I really just want to discuss the glands that come into play, such as the seminal gland and we'll get to the prostate, just the seminal gland for now. And so the seminal gland is what's going to secrete the thing that we need to produce semen, right? It's going to produce 60% of the volume. And just as an FYI, again, I mentioned that the testes, what is viable sperm, like sperm cells, only 10% of it is viable sperm cells that can fertilize an egg. But the rest of the content that comes out, the ejaculant that comes out during male climax, is pretty much substances to ensure the success of sperm in its journey to find an egg. And one of those things is to stimulate the flagellum to move. What is the flagellum? The tail. That is the correct technical term for the tail of sperm cells. To stimulate, the flagellum is the technical term. And so you can see that 60% of our volume is now going to be contents from the seminal gland. And one of those major things that it does is that it stimulates the flagellum. And the funny thing is, just if you wanted to know, it's actually just fructose. Fructose, a sugar. Sugar is what actually makes it move. Now the next gland, when we go back to this image here, our hand, you don't have to flip pages, I have it here for you. Now we've reached here. This is our vas deferens. You can see sperm has made its way here and it's stored in this whole ductus deferens as fully mature sperm. Right here is where they are stored until they fully mature. But here they finally fully matured and they're stored. And when they need to be released, they're going to go through. And you will see now you have your seminal gland. And this seminal gland will release that fructose that the flagellum need to actually carry out the whipping-like structure. There is one term that's cut off from here. There is an ampulla here. This is where sperm is primarily stored. It's not the entire length. It's actually more along here, but it's okay. I'm not removing it. It's just something I want you to be aware of. And so we've now made our way into our next gland, which is going to be our prostate, and it produces the last little bit we need of our semen volume. And what it contains is something we refer to as seminal plasm, which is an antibiotic to prevent an infection in the urinary tract. So it protects this urinary tract from infection. And that's what this gland does, the prostate gland. Prostate gland. It also does contribute other milky alkaline substances, which I'll write it into for us in a second. But that milky alkaline substances, if you're not familiar with the word alkaline substances, spelled like this, alkaline, this means like a base, and we need a base because if this is acidic, sorry, let me get a better color. If this is acidic, right, because of all the urine that travels through here, it needs the sperm needs protection right before that space. Let me make this larger. Right before that space, and so the prostate gland itself contributes to those proteins that protect from the acidity of the urethra. So that's what this prostate gland is known for. And it also releases an antibiotic, again, to protect this urethra from infection. So it does those two functions. I'm going to write them both in. Seminal plasms are our antibiotics. And our second function, alkaline mucus. Which is a base, helps neutralize acidity in the urethra. Neutralize acidity in urethra. And next, we finally made it to our final gland, and this is also going to secrete that thick alkaline mucus that we need to protect it from the urethra. Neutralizes acids in the urethra, but it also occurs in the prostate. The prostate does the same thing. The last thing to add, we mentioned it a little bit earlier, it's that last gland that lubricates the tip of the penis. So some functions of the penis is to conduct urine. This is what its technical definition is, to conduct urine, and to introduce semen into the female vagina. Its biological function. Now, as we go a little bit further out, that's everything that's small that matters for our journey out, right? We started here. We had left off here. We're here traveling through the prostate. We now made it through the urethra. And now here you can see bulbal urethral gland. This also used to be called Cowper's gland. You may see like that in your medical textbooks in your next advanced professional school. And so this used to be Cowper's gland, but it's also known as bulbal urethral gland. And again, it secretes that alkaline substance to protect from acidity, just like the prostate. And it also lubricates the tip of the penis. Now, when you look at the overall structure of the penis in general, the portion that is not exposed or within the bone, you can see here's your obturator foramen, here's the pubic bone, here's your pubic symphysis, and here's a little bit of that other pubic bone. And so you can see the part of the penis that is within that pubic bone or within the pelvic bone is going to be considered the root of the penis. And then the major body of it is just called the body of the penis. And then the head is not called the head. I know that even in colloquially we call it the head, but now you should know that what everyone always refers to as typically the tip or the head is actually technically called the glans penis. That is the proper term for it, glans penis. And the segment right before there's a rise, like a little bit of an edge, is called the neck. Called the neck here, the neck. Just that segment that where there's a little bit of an elevation going into the glans penis. All right. And now the erectile tissue. If you were looking at this and you were to do a transverse section and you were to look straight inside, like straight looking at it, what you would see is this very image here. And what we see are three compartments, two that share the name. First, this segment up top is going to be your, my highlighter, corpora cavernosa is going to be the first one. That's going to be the segment up top. And then the segment on the very bottom is going to be your corpora spongiosum. Corpora spongiosum. And you'll notice that they have a word that sounds familiar, like spongy, to be like sort of have a lot of crevices, have a lot of cavern system. And so if without giving names to things, what happens is that this segment, the corpora cavernosa, is responsible for the engorgement of the penis. There are blood vessels that will fill that space with blood, and that's why the penis goes from flaccid to being erect, is because it fills up with blood, or it fills the corpora cavernosa with blood. This other segment, corpora spongiosum, is meant to be more protective. This is the urethra where urine and sperm will be traveling through. And so this layer is responsible for making sure that it doesn't seal shut, right? So that this pathway is open. So even if there's some downward pressure during the act of sexual reproduction, it still allows it to stay open so that sperm can make its way out. Anything else I want to add here? That's really it. Be able to identify which ones, which labels, or which parts which. And remember, this is the segment that engorges with blood. This one protects the urethra. That's their easy functionalities, no matter how anything's phrased. The spongy urethra protects the urethral passageway, and the corpora cavernosa is responsible for being engorged with blood. Just like when you're looking at when you're looking at, say, a shark or a fish, that fin that's on the top, like when you look at a fish swimming on, a shark swimming on the top of water, it has a fin that sticks out. That's the dorsal fin. And so, sort of in that same spirit, on the superior portion, you would encounter dorsal veins and arteries and nerves at the very top. And they're eventually going to innervate and serve the rest of it. But that's where the name comes from. Everything else seems a little bit extra to me. I really just care for you to know what the functions of those two in bold here are. And that's really it. And your notes are just nicely complete so you can have it. Descriptors. And so this is another picture of us. This is a superior / posterior view of things. And it's the same image as you're looking at here. Here was the testes, here's the epididymis, and then the ductus deferens. They remove the urinary bladder. And then you can see this little segment. This is where sperm is primarily stored until it's ready to be released. And then the seminal gland is the one that contributes some contents of semen. The prostate contributes the other 20 to 30% of semen. And then the bulbourethra is the last bit that contributes that last bit of semen and also lubricates the tip of the penis. And so you're seeing the same image as before. And if you look at this passageway, here's our urethra. And so the surrounding tissue around it is going to be your spongy your spongy body. That's what this word means in Latin. Corpus means body, spongiosum means spongy looking, and so the spongy body. But corpus spongiosum is the correct term. And so surrounds the urethra. And then outside of the urethra, you have your corpora cavernosa. So it's just to see the same image or the same terms in a different from a different perspective. And again, that's going to be responsible for the engorgement of the penis. Keep in mind that the root of the penis is inside before, like inside of the pelvic bone. So this is, you're looking at a boundary as you look at the root of the penis where this label's at. So you can see that the bulbourethral is within that pelvic bone, the prostate, the urinary bladder, all of this within that pelvic cavity. And another word to add here, on the glans penis, on the tip or the head of the penis, the glans penis, there is a piece of skin that overlaps or covers it entirely, and it's called the foreskin and the prepuce. And the general functionality is to sort of keep it in its own juices, such as the bulbourethral secretions. Those bulbourethral secretions are responsible for ensuring that the tip stays healthy or protect you from bacterial infections. And so the foreskin helps trap a lot of that fluid that serves as an antimicrobial function. And so, and then in our general culture, this is the part that gets removed. And there's no general differences, or most people won't really suffer from too much, but you may be prone to certain types of infections or particular types of bacterial species if you don't have it. But otherwise, for the most part, proper hygiene is enough to keep you healthy and safe. Oh, and last thing, so this question came up twice earlier this week in because this sort of always serves as, sex ed for some students. I know that you may not believe that, but it is. And a question that I got twice, and I get it every year, is when a male becomes aroused and something comes out in the very beginning, that's the bulbourethral gland secretions, and that will not actually get you, that is not capable of fertilizing an egg. And it's a common question, is like, I believe the colloquial comment, I know it's probably a little unprofessional, but I'd rather just you guys be educated in this stuff. The pre-something, that's what comes out of, that cannot fertilize an egg. But the dangerous thing is that if there's been sexual activity beforehand, this bulbourethral gland can definitely push out viable sperm cells onto the tip or the glans penis, and that can cause fertilization. And so that's just a little bit of FYI. So be very careful in your lives. And so, yes, use protection. Condoms is always the safest thing to use. And so with that, in my sex ed part, let's move on. So now the semen. Again, do not forget that semen contains sperm, and really seminal fluid is another word for it. Honestly, they're interchangeable to me. I think that this textbook defines semen possesses sperm and seminal fluid. So when we were talking about before, we were saying, 30% from the prostate gland, 10% bulbourethral gland, 10% sperm. Overall, the terms are used a little bit differently than when I remember being taught, or either I learned it wrong. So I'm just going to clarify what's modern. And so to correct what I said or clarify what I say, semen contains sperm, which again was 10%. And then all the seminal fluid given by the seminal glands, given by the prostate gland, the bulbourethral gland, as we discussed a little bit earlier, right? Seminal glands, the prostate gland, and the bulbourethral, it's the only ones we've talked about. We don't go that deep into the epididymis. But all of these glands these other two cells that you're seeing here will also be capable of contributing something to what comes out with sperm during male climax, with the ejaculant. And so typically everything that is not sperm is considered seminal fluid. But you can see how it can be confusing, because seminal fluid, I was taught seminal fluid comes from seminal glands. And then the prostate glands drops off its own things, prostaglandins. And so this textbook, I either have to talk to the publisher or maybe my knowledge is wrong. So just keep an eye out for that, because that's something I've noticed. But as far as we're concerned, Semen is going to be made of sperm and fluid, okay, our fluid. And let's keep it with what I said earlier. I think I just prefer it this way. So you can see 60% semen volume. And so the prostate contributes 30% semen volume. And so the publisher wasn't very clear on this, and I never contacted them between last semester and this semester. But otherwise, this feels a little confusing. So what I'm going to do, and I hope you're aware of this for the future in case I'm wrong, we're just going to call 10% is sperm and the last 90% is semen. The same way all your notes are reflected, just for the sake of simplicity. And it's approximately around 2 to 5 milliliters of what comes out of ejaculate. Now, last thing, when you enter physio, the first thing you're going to do is go back into cell biology. You're going to go into how do things move across the membranes, because it's going to feel, physiology is honestly feels to me like more of a cell biology class more than it does anything else. And so one thing that they're going to intro in is the difference between mitosis and meiosis. We have never discussed meiosis, so you should know what mitosis is simply by looking at this image. One cell will develop 2 daughter cells. That's it. And then one will be destined to stay and one might move. We learned it more easily with skin, that you have the basal layer at the very bottom of the epidermis, which would give rise to all those little squamous cells, those keratinocytes that are going to go to the surface of your skin. And one would go up, one would stay, and then again, this one would repeat the process over and over again, creating another one, and one stays. And so these, we've always been calling them in the class as the stem cell population, the stem cells. So it really means making a copy. This makes an exact copy. You can even look at the chromosomes of the DNA organization, black, red, blue, a double there, a double here, double there, of all the same exact colors. They are identical clones. That's what mitosis is for every single cell in your body. And you've learned about it very slowly and generally. For example, an osteoblast, an osteocyte come from an osteoprogenitor cell. That osteoprogenitor cell is an example of a cell that undergoes mitosis, creates a copy of itself, and then it differentiates. It turns into a osteoblast, and then so it works like what's that, the little game, the anime game? Pokemon. Like one goes into one and it evolves into something else. When we're looking at mitosis, you are pretty much thinking about just it making an exact copy of itself. Whenever that cell was turning into something entirely different, it was called differentiating, like kind of like evolving in that context. And so now, what the heck is meiosis? One important note that's important for me to leave this class is that meiosis only occurs in sex cells, aka, that doesn't make sense, aka eggs and sperm. Every other cell in your body does not undergo meiosis. Meiosis only happens in eggs and sperm. And I'm going to give you a generality as to why. When you look at the end product, mitosis only really wanted to generate a copy of itself. That's it. That's mitosis. There's nothing else to it. You would go through all your phases of mitosis. What was our acronym for mitosis? Anybody remember? Oh. IPMAT, exactly. Right? Interphase, prophase, metaphase, anaphase, telophase, and then you end up with 2 cells. That's the process of mitosis. Now, what is meiosis? It is using these same processes, but the overall goal is not to end up with a copy of itself, but to end up with 4 unique little instances of what it was. Let me say that one more time. The overall goal at the end of the day of meiosis is to produce cells that have a mix of the genes, a mix of mom and dad in the sperm cells. In other words, if you're a male and you have functional sperm right now, mom and dad's genes that make you, every single one of your cells has the instructions of dad and has the instructions of mom. But when you're making sperm cells, if you're a male, what it's doing is that it's scrambling them all up and mixing them up evenly so that you get 4 different variations of genetic content. And so just one cell will eventually lead to 4 functional sperm cells. 4 functional sperm cells. And so that's a big difference between meiosis and mitosis, is that when you hear the mitosis, we're just making a clone. You can even see the chromosomes are evenly distributed, just like they were when they started. But if we look at the beginning, they duplicate them. They make a bunch of copies of it. They start undergoing mitosis. Mitosis is, again, these steps here: PMAT, prophase, metaphase, anaphase, telophase. And so they undergo PMAT. These two, this one will undergo PMAT again, and this one will undergo PMAT again, and then you end up with 4 sperm cells. These will eventually mature. So this is what I mean by immature sperm cells. Cells that are eventually destined to differentiate into sperm cells, but they haven't developed their tails yet. Remember that the tails develop in the epididymis, and so based on our knowledge right now, where is all of this occurring? Where is meiosis occurring then? When we look at this picture right here, where is it occurring? It's occurring in our seminiferous tubules. In our seminiferous tubules, all of these cells are undergoing meiosis and developing functional sperm cells that will rise and leave this space. But again, meiosis only occurs in the testes of males and in the ovaries of females. But otherwise, meiosis only happens to sex cells. Now, I don't want you to know the processes of meiosis and mitosis. I'll let physio traumatize you with that. What I want you to know is meiosis only occurs in sex cells. I want you to learn this vocab word. Another word for sex cells is gametes. And I'm just trying to set you up really well for the beginning of physiology. Gametes. Like game and then tes. Gametes. And so we refer to eggs and sperm as the gametes. Sperm is the gametes of males. Eggs is the gametes of females. I hope we know how to use that word. And we'll leave it at that. And so it's just important for me to know the difference between where meiosis occurs and mitosis. That mitosis generally is just making clones. Clones, that's it, exact copies. Meiosis is not about making clones. It's about ending up with 4 individual different little sperm cells. And that's really it. So don't let all this intimidate you. It's really just to intro you to something that's going to be more complex later. Now, where it matters and what matters now is this image I always found very confusing unless people understood what was going on between these steps. That you have one cell that's always there dividing into, you have one cell that's always there dividing. It divides into one that needs to turn or differentiate into something else. And so we'll undergo meiosis 1, meiosis 2, and then these will mature into functional sperm cells. This is occurring in the testes, in the seminiferous tubules. And that's what you're seeing when you look at this image here. You are seeing the beginning, and then it is undergoing meiosis 1, meiosis 2, and then finally we end up with functional sperm cells. Along the edges of those seminiferous tubules exist all the cells that will eventually become sperm cells. They create one that will eventually go down and start dividing to form gametes, functional sperm cells. And this is occurring once more in the seminiferous tubules, in these little tubules. And so I will leave it at that, all right, for the testes and meiosis. And I guess I really don't have much to ask. I should have labeled it up here if I really wanted it to. Oh, it is. But it's okay. Honestly, I'm going to keep this. This is something I want you all to highlight. I really just want you to know meiosis, where it occurs, and that's it. That's all. I can sleep at night when you go to physio with that. And we can remove nerve cells and epididymis. Just, I think that's confusing to add it to that segment here. Just cross that out. It's okay. We won't need it. And then it'll have a little bit more overlap with lab and nothing extra to lab. It'll be exactly like what lab covers. All right. So now moving on to the female reproductive system. And so big differences at a glance without giving names to things. We already know some of them. Like, for example, one thing that I want you to note is that males, remember that they had two exits, right? They had anus and then they had the urethra that comes out from the penis, right? The opening from the penis. And here, the anus is going to be where feces, rectum, right? We learned about this in lab today, Thursday's lab. You'll learn about it in lecture. Lab next week. This Thursday. And so this is where feces will exit from. And then there'll be two openings. One will be similar to the males. It'll be the urethra. And this is our urinary bladder that we're seeing, just like males. But the big difference is that females obviously do not have a penis. And so that's the big difference, because we notice now that the vagina is going to be another separate opening. So you can see that urinary and reproductive in females is separate, as opposed to males, they share a pathway. And semen is dependent on, the role of semen is to pretty much deal with that very setup, right? To neutralize those acids from urine. And so with that. And let me see what pictures. Here it is, sorry. And so when we're looking at this image here, this is going to be looking at it, the perineal space, right? The space that's between sort of the anus and the reproductive organ. And now this entire segment here is considered the external genitalia. I know that we've always considered the female reproductive system to be an internal structure, but it has structures on the outside. Altogether, everything that you're seeing here is called the vulva. The vulva. The vulva describes this entire region that I'm highlighting now. So some structures. At the very top, there is this particular segment called the mons pubis on the underneath this skin layer that actually stores a little bit of fat that's always there. It just happens to have this little storage location for fat called mons pubis. It has a prepuce. This is the same word as the word foreskin, and the foreskin covers, you write it in, foreskin covers, and it covers the clitoris, and the clitoris's role is in arousal. And then vestibule. This word vestibule always tends to mean opening or where there's going to be an opening. So you'll see this word, we've probably seen this word already come up a few times in anatomy. So we're just going to leave it as opening. And then we have a minor lip and a major lip. Labia is the Latin word for lip. And again, lip here. Labia, lip, and then minora for minor and majora for major. And so the most superficial one is going to be labia majora, and the one that's inferior, closer to the vaginal opening, is going to be the labia minora. And the hymen would be here. It would be a piece of connective tissue that's web-like before being lost after the first sexual encounter, or sometimes through rigorous physical activity such as horseback riding, gymnastics. That connective tissue can rupture. And anything I'm missing here? And glands. And so there are glands just like in the male system that were responsible for providing lubrication. And so you have the bulb of the vestibule and the greater vestibular glands. They are two separate glands. All responsible for lubrication, either inside or outside. And so if we look at our definitions, this one will activate on the outside. Will activate into the outside, into the opening, right? Greater vestibule. Sorry, let me get my highlighter. Greater vestibule. will discharge into the vestibule, into the opening, right? The outside, what's denoted by these arrows here. Yes. And then the vestibular bulb. Now, it has always been commonly known that males, when they're aroused, right, we know that the corpora cavernosa, we know that it fills, engorges with blood, and believe it or not, the clitoris is actually a miniaturized version of the penis. It actually has the same setup. It has a corpora cavernosa, and so it has that spongy looking tissue that allows for blood to sort of fill in that space, and it actually, during arousal, will swell up or become erect. And so the vestibular bulb is the one responsible for I don't know why I didn't write it, but they don't have it written here. Is going to be for, I'm just going to put, we're going to put it synonymous to what the corpora spongiosum does. This is all just wordy text just to really just say one thing, that it does exactly what the corpora spongiosum does. Is that sort of, it's a erectile tissue. That's really just the best way definition for it. The erectile tissue. All right, and I think we described them all. And lesser, I'm not seeing on your image or diagram. So I suppose we won't have to worry about it. Yeah, there's one more gland. It's not in your image. It's a little disappointing, and it sits underneath this bulb of the vestibule. But so go ahead and just cross this one out. Since we took off some terms for men, we're going to take off some terms for the female as well. And next semester, I'll change my images. All right. And so that covers our external genitalia. So now we're going to go internal. And so, as we look at this image now, here is our labia, here's the majora, here's the minora, right? The major lip, the minor lip, and then here's the vagina. And then you can see some accessory glands, like the ones we just learned about, the vestibular gland and the bulb of the vestibule. And so these are the images, and you can see here's our lesser one. This is a picture of the lesser one, so you can see it's a little bit deeper, so you're not going to see it from an external view, like looking at the vulva from that direction. And so the lesser one can't be seen, but don't worry about that term. But otherwise now, if we make our way into the vagina now, we're traveling, and then you're going to see this object here that makes an upside down U as you're sort of seeing. Let that remind you of the name of this organ. It is called the uterus. This entire segment is called the uterus. Uterus. This uterus, its function is to be prepared for a fertilized egg finally arriving to this uterus. Without using terminologies, just to explain what actually happens here, this is the site, much like the testes that produce sperm cells, this will produce the eggs. Those eggs will be released during ovulation. We'll explain what that means in a bit, or probably next class. During ovulation, the egg will be released into the fallopian tube, which is also called the uterine tube on your notes. Uterine tube, fallopian tube, it's the same tube. And this tube, the egg will make its way, and then in something we call the ampulla, which we'll discuss in detail in a second, it hopes that a sperm cell will finally arrive and find it, and then finally fertilize that egg. And if that egg is fertilized, it will travel, and eventually end up here on this inner lining. And much like an alien movie, if this egg is fertilized, this layer is so vascular. There's blood vessels just rising, and then when a fertilized egg reaches that location, they like an alien movie, they innervate with it. And then they sort of form a union with it, and that's how the fetus will eventually become sort of one with the womb, right? One with the mother. And so this layer has a very specific role in catching fertilized eggs. So if it catches a fertilized egg, it begins the process, or what we know as pregnancy, or the development of that fetus. A little bit more on that a little bit later. And now, if that egg is not fertilized, if that egg arrives here and it's not fertilized between day 15 and day 30 of after ovulation of the menstrual cycle, then this egg will be removed from the uterus through excretions, which is for the discharge that leaves during the menstrual cycle. So that all that blood is usually described as typically as mucousy in appearance, and that's because you're shedding the inner lining of this uterus, as you can see in white here, and it's getting rid of that unfertilized egg. Because there is one fundamental difference to understand between males and females, is that these things are always working. They don't care about what time of the month it is. The testes are always producing viable sperm and resupplying all the way until old age. And in the female system, there is only one egg every month that will be a contender for fertilization. And if it is not fertilized, then the role of the uterus is to get rid of it for the next month to allow a brand new egg to have the chance of being fertilized. And so it's a big difference between males and females, just biologically speaking, or the rate of the development of the gametes of the sex cells. And so with that, now the words in bold here describe ligaments that matter. When we look at this structure here, you're looking at the same organ. You're looking at a uterus, you're looking at the fallopian tube or the uterine tube, you're looking at an ovary, and you're even looking at the vaginal canal here. You're just looking at it from a this is posterior view. So this is from the back end, posterior view. Normally you wouldn't be able to see it anterior because you have your urinary bladder sitting here. So you wouldn't be able to see it from the front unless it was removed. That's why I kind of just thought they removed the urinary bladder, but they're looking at it from a posterior view. And so with that, here we encounter the vagina, right? The entrance into the uterus, the boundary around it is called the cervix. The boundary, like if you're playing basketball and there's a rim, the rim is going to be the cervix. The opening is going to be called the external O's. External O's, that opening inside. The ligaments I only care for you to know are typically the ones in bold, so I know there's a lot of extra terms here, but otherwise the ones that are in bold are what matter the most. Everything in bold is always what will matter the most. And now the ovarian ligament, if we look at our ovary, there is a ligament that attaches it to the uterus. That attaches it to the uterus. You have another ligament that's very broad and large called the broad ligament, which attaches the uterus to the abdominal pelvic cavity, right? More so the pelvic cavity. And so that's what the broad ligament is. And any other ligaments? Which other ones do you have? Mesovarian and broad, suspensory. And then we have our suspensory ligaments that anchors the ovary to the body cavity. So that's the real difference between ovarian. It anchors it to the uterus. And then we have our ovarian ligament, which anchors it to the body cavity. And finally, mesovarian. Just like mesentery, right? Mesentery, we're going to hear, Thursday lab, you may not know what mesentery is yet, but mesentery is going to be a connective tissue sheath that allows for blood vessels to come and innervate into spaces. Mesovarium. Mesovarium. And this is a big comparison, and do not get intimidated by this. It's just going to be simple definitions. This is the same box as our meiosis and mitosis box for sperm cells. And mitosis, again, nothing changes. When a cell undergoes mitosis, it's just making one copy, an exact copy of itself. But when that egg is getting prepared to be released, or it has to be released, it cannot send a complete egg, right? The stem cell population. It cannot send it as is. Because in here, you have the complete chromosomes to make a human being. The complete chromosome that make a complete human being. Like if you were to look at this and take this out, and you were to compare it to a skin cell, like let's say this is all your mom's, right? Your mom's egg cell. You take it out, and if you were to compare it this at this point, and you were to compare it to any cell, the DNA looks exactly the same. All the chromosomes are exactly the same. All the genetic content exactly the same. But you can't have that to make a functional, viable egg. Why? Because to make a human being, you need half of mom, half of dad. So this is too many chromosomes. Too many chromosomes. So the role of meiosis has always been, even when we were evaluating it in sperm, the role is to not be have the complete genetic content of a male and female, mom and dad, but to mix them up so that you have half. So these sperm cells will have essentially half of the genetic content of what every single cell in your body has. And that's why they're different types of cells. Because they're not like the rest of the cells in the body, because they don't contain the same genetic information. They contain only a mix of mom and dad, but not all of it, because you cannot have more chromosomes. So these chromosomes together, when they combine with, dramatic effect, here when they mix with your oocyte, they have to have just the half of the genetic content so that when they come together, they will be able to finally have the right amount of matching pairs of chromosomes, and they will make a brand new human being mixing mom and dad. And so that's sort of the general premise. So nothing really changes as to what I want you to know. We're keeping it very simple for our class. Meiosis is about creating gametes or sex cells. It doesn't change. And one little interesting fact I really want you all to just leave here, and I'm not going to test you on, it's just for you to know. When you look at this, you start off with 1 and you end up with 4. So one functional spermatocyte, a cell that it's eventually going to become sperm cells, will lead to 4 functional sperm cells. With the female system, it is far away from that setup. You can see that 1 will eventually give rise to, yes, 4. Here's 1, 2, 3, 4. But the unique thing about the female system is that it only makes 1 viable egg. So instead of making 4 viable sperms, the actual female gametes will go from 1 cell to creating 4 cells, but only one of them is useful in fertilization or capable of being fertilized. And so that's what this image denotes here. This is a functional egg which found a little sperm cell, and now these chromosomes are going to work together, line up together, and form a functional person or a functional fetus inside of the womb. But then what happens to the rest of it? These are called polar bodies. Their functions are not that well known yet, but I believe that one of the first key findings that we have about it is that polar bodies contribute by adding or giving proteins and any necessary materials to the egg as necessary. So a lot of times you'll see polar bodies sort of stuck to the oocyte like this, and they're sort of providing contents for that egg. And so that was the last thing I learned about in my graduate studies. That was the last thing that they kind of knew about it. And there's not really enough time to keep going, so the last thing I just want to add, we'll be done pretty quick,